Hi everyone, this is Luth. Today I will be covering enzyme kinetics. We will start off with the Michaelis-Menten model, graph, and then the equation. And right after that, I will teach you how to convert the Michaelis-Menten equation to a Lineweaver-Burk plot. So firstly, what are enzyme kinetics? Now, enzyme kinetics is the study of the enzyme catalyzed reaction. Enzyme kinetics can be elucidated by assuming the simple case of one substrate and one product. Okay, first let's start off with the Michaelis-Menten model. So I'm going to draw two axes right over here. Okay, and the y-axis is for your velocity and the x-axis is your substrate concentration. Let's say I want to start the reaction, so your velocity and your substrate concentration is 0. Now, as I add more substrate into, for example, I have a test tube filled with enzymes, and what I'm doing is I'm adding more and more substrate into the test tube, which has enzymes. So now I'm adding and adding and adding. So I add and add and add the substrate, and at a point of time, it will just become a horizontal line. What does this mean? The reaction has now reached terminal velocity. But why does it reach terminal velocity? Now here's why. For example, I have a test tube with 10 enzymes and I add 10 substrate. So now all the enzymes are occupied by a substrate, and then I add 20 substrate and enzymes are still 10. So all the enzymes that are in the test tube are all saturated, which means it has a substrate with it. It's an enzyme substrate complex. So for example, if all of the enzymes are saturated, no matter how much substrate you add in, no matter how much further you add in, it will still be the same. The speed of reaction will be the same, because the speed of the reaction is controlled by the number of enzymes present. So the more enzymes there are, the faster the reaction will go. But at a point of time when your substrate concentration exceeds the number of enzyme, it will eventually reach terminal velocity. So you have to know that. So in this graph, let's analyze this graph. So right here, where it reaches terminal velocity, it is Vmax, maximum velocity. How do we get Km? Do you guys know what Km is? Km is the Michaelis-Menten constant. It is the concentration of substrate when V0 is equals to 1/2 Vmax. How to get Km? By saying the Michaelis-Menten graph. Firstly, you take your Vmax and you take half of your Vmax, which is Vmax over 2. So Vmax over 2, you read it off in the x-axis, so you read off the coordinates on the x-axis, and this coordinate right here is Km. Km is the Michaelis-Menten constant, where the concentration of substrate when V0 is equals to 1/2 Vmax. So this Km, what does this Km mean? How to interpret the value of this Km? So if the Km is high, a high Km, so this means that it has weak substrate enzyme binding. The enzyme has low affinity for the substrate, and the enzyme substrate complex will dissociate. Basically, the enzyme don't like to be with the substrate. But if the Km is low, it means that the substrate enzyme binding is strong, and the enzyme has high affinity for the substrate, and therefore it forms your enzyme substrate complex, and the reaction will proceed forward to form your enzyme and your product. Now for this particular reaction, for example, how to calculate the velocity of this reaction? Now V0 is equals to Vmax multiplied by your substrate concentration over your Michaelis-Menten constant plus your substrate concentration. There you go. So this is how you calculate the velocity of the reaction. But then you look at this graph, it's a curved graph, it's not a straight line graph, and scientists, we don't like to use curved graph, we love to use straight line graph because it's easier to read out the values when the graph is straight. So these two guys, Lineweaver and Burk, they decided to straighten this graph, to turn this graph into a straight line graph by doing a double reciprocal of the Lineweaver-Burk plot. So the double reciprocal Lineweaver-Burk plot, is a linear transformation of the Michaelis-Menten plot. Alright, so this formula is given by 1 over V0 is equals to Km over Vmax multiplied by 1 over substrate concentration plus 1 over Vmax. Now you tell me, you have to memorize this formula for the exams. You have this to memorize, which is your velocity in your Michaelis-Menten model, and you have to memorize your this equation for your Lineweaver-Burk plot. Isn't it irritating? Why? So in this tutorial, I'm going to teach you how to derive this thing. For my exams, I'm not tested on both formulas because they are both given to me, but I took the extra effort to know how to derive the formulas, and it really helped me in my exams. For biochemistry, I scored an A. It's because I go an extra mile. So in biochemistry, my tip to you is to go an extra mile to do a little bit more research so that you know what you're doing. Because in biochemistry, if you're blindly memorizing, you don't know what you're doing, most probably you'll fail or you won't pass it good, and because you don't have passion for the subject. Anyway, back to the topic. So let's start with your first we have to start off with our Michaelis-Menten equation. As I said just now, V0 is equals to Vmax multiplied by your substrate concentration over Km, which is our Michaelis-Menten constant, plus the substrate concentration. And from this equation, we're going to transform it into the Lineweaver-Burk plot equation. So the transformation involves double reciprocal, which basically means it's more mathematical instead of scientific. So your algebra and your fractions and your manipulation of your terms must be good. But it's really simple, actually. It's really simple. It's kind of like high school maths. So let's start by doing a reciprocal. So first step is to oh my god. First step is to do a reciprocal. Okay? Reciprocal. So how do you do a reciprocal? You just invert everything. So this is V0 over 1. Is 1 over V0 equals to Km plus substrate concentration over Vmax multiplied by your substrate concentration. So basically what you're doing is doing a reciprocal, which means you're just inversing the fractions. Okay, I don't have space here, so I'm just going to do the same thing on the other side. So 1 over V0 equals to, so this is basically what's on the previous slide. So once you have done this, what you're going to do is we are going to separate this into two different terms. So next step is to separate. Okay? Separate. So your 1 over V0 is still the same. Now you separate this whole term. So it becomes Km over Vmax S plus S over Vmax S. Now what you can see is both of these cancel each other out because they're in product form. So now we have 1 here. Alright? And then after that, what we do is for this part, we separate this part. So how do we do that? So this still remains 1 over V0. Km over Vmax. So this is basically multiplied by 1 over S. And plus 1 over Vmax. And guess what? This is the equation. So guess what? That's it. You got to derive the equation. Don't believe me? This is the equation I showed you earlier. This one. This whole thing. You just learn how to derive it right here. There you go. This is your Lineweaver-Burk plot. And also, if you can notice, this is in the form of y equals to mx plus c. So this is how the graph looks like. It looks like this. And it goes like this. So your y-axis is 1 over V0, your y-axis, your x-axis is 1 over substrate concentration, your slope over here, your gradient is Km over Vmax, and your y-intercept, which is your c, is 1 over Vmax. So that's how you interpret the graph. So let's just do a little bit of recap. So first we start off with your Michaelis-Menten constant right over here. And then we do a reciprocal. So basically we invert everything. Just normal inversion. After that, we separate this term, we separate this whole thing to get this. After we separate, we cancel off the S, which is a common thing in your fraction, so we just cancel it off. And then after that, we separated this part. We separated it into two terms, and there you go, you derived the Lineweaver-Burk plot equation. And then from that, you can look at this trend, this graph, this equation fits into y equals to mx plus c, which is a straight line graph. So if you see Lineweaver-Burk plot, that's how you interpret the equation. How do you interpret the graph based on this? So this is a very important this is very important on how you actually transform it, because most people don't know it, but it's so useful. It will help you a lot and you will understand it better. Okay, so this is part 1 of biochemistry for enzyme kinetics. In the next tutorial, I will be doing enzyme inhibition, where I will cover irreversible and reversible inhibition. And in reversible inhibition, there are competitive, non-competitive, and uncompetitive inhibition. And I guess that's all for now. If you do have any questions, please do leave me a comment below. I will help you as soon as possible. I really do have people asking me questions and I've been helping them. So ask me questions if you do have any questions. This is my favorite topic. This is my favorite module. Biochemistry is so my thing. So if you do have any questions, just let me know and I'll help you as much as I can and as fast as I can. This is Loth, you're watching PsyCrafties, and don't forget to give me a thumbs up. And if you found this helpful and you want to be notified of my other videos, don't forget to subscribe to my channel. See you next week.